Okay, let's get into it. So I want to talk about the history of how we have arrived at our current understanding of basic microbiology. So one of the things that we think of now, one of the things we know is the cell theory. This is the fundamental assumption we make when we think about a lot of biology. And this just says that all living organisms are made of cells. In microbiology, we think of a lot of single-celled organisms, but they are cells. As you've learned anatomy and physiology, you've learned about the cells that make up a human, and each one of those cells comes from another cell. So another cell goes through, through cell division to make a daughter cell. And every single cell in a person's body had a mother cell, and that cell had a mother cell, and that cell had a mother cell, and they can all trace their lineage back to the fertilized egg. And that fertilized egg can trace its lineage back ultimately to the fertilized egg of both of its parents. So that's the idea that all cells come from other cells, and that is a huge idea. 200 years ago, people did not think this at all. This was a revolution in our understanding of biology. So how did we get to the cell theory, and where did we start? That's one thing we're going to look at. Another thing we're going to think about is the germ theory of disease, and this is our modern understanding of infectious diseases. It's a scientific theory, just like the cell theory, and it says that infectious diseases are caused by microorganisms. This also was a revolution in our understanding of medicine. And so we will look at how we got to the germ theory of disease. And again, when I say theory, I'm talking about a scientific theory. That's a testable hypothesis that has been tested many times and has never been disproven. So the germ theory of disease has been tested again and again and again. At first directly, and then later as a byproduct of many experiments. So initially, long before we knew what microorganisms were, and eventually once we knew what they were, but long before we understood how they worked, a very common idea was spontaneous generation. And this was the idea that living organisms can come from non-living things. So if we found bacteria, we could imagine that they did not have a mother cell. Instead, they spontaneously appeared. That's spontaneous generation. So something like flies that would appear on rotting meat, they would appear. I'll explain what this is about in a little bit, but spontaneous generation was where the idea of Frankenstein's monster came from. It's the idea that there are these forces that we don't understand very well, and if we could understand these forces, we could create life. And so in the 1800s, this vital force was romanticized. Once people started learning about electricity in the 1700s, they thought, well, electricity must have something to do with the vital animating force. And now we know about things like nerves and action potentials, and we know about things like electron transport chains in cells. These all involve things like electricity. But you can't take a dead thing and put a vital force in it and make it alive. You can't build Frankenstein's monster using any kind of blunt things like electricity. Miasma, we will talk about a little bit more than other things, because this was one of the last accepted ideas for what could create life. Miasma is pollution or bad air or corrupting gases, and it can if this existed, what it would do would be cause disease. So if you smelled something very bad, that bad gas would give you a disease. That's miasma. And this was one of the best theories we had to explain how infectious diseases happened. As recently as the late 1800s, this was the best idea people had, and this is one of the last one of the last theories to be disproven. So, can you disprove spontaneous generation? Can you think of something in your life where you could see a new cell or a new animal and prove that it came only from a mother cell? And in particular, can you prove that it was impossible for that thing to be created in any other way? So just go ahead and pause this and think about it for a while. 3, 2, 1, pause. I presume you would have seen some like animals being born, or you might have seen a plant grow from a seed. And those are things you witnessed, but that's not the same thing as disproving the whole idea that a vital force or miasma could create life. And scientists for many hundreds of years tried to, at a very basic level, disprove the idea, well, prove or disprove the idea that non-living things could become living. So one of them was Francesco Redi. He had observed maggots growing on rotting meat. So this was in the 1600s, that he knew of a thing that would happen if meat was left out, it would rot and maggots would appear. And he thought maggots did not arise spontaneously. But no one knew what maggots came from. So some people thought they would just spontaneously appear. So he generated a hypothesis, which was maggots arise from flies. He knew, and other people knew, that maggots become flies, but they didn't know where the maggots came from originally. So his hypothesis was that flies make maggots, and then maggots become flies, and that is a cycle. So he designed an experiment to test this. And what he did was get some fresh meat and put some meat in 2 different jars, and he would change a variable between them. One of them would have a cloth covering and one of them would be open. And his idea was that the open jar, flies could enter it and create maggots, and the covered jar, flies could not enter. They could not create maggots. And that was the result he found. Maggots could only appear in the open container. And his conclusion was that flies were necessary for making new flies. Some things I want to point out about this are that the open container, so the 2 jars, it was necessary to have 2 jars in that experiment. And I want you to think about what would happen if he had only had 1 jar with the covering on it and no maggots had appeared. Why, if this hadn't been here, why wouldn't this be enough? Go ahead and pause the video. So what I'm getting at is that his rivals could have said, oh, if all you have is this jar and no maggots appeared, there was something wrong with the meat you used and no maggots would appear on that meat. Or there was something wrong with the jar and it killed the maggots. There would be any number of different explanations a person could have for why no maggots would appear. But since he had both jars, since he had this control that allowed maggots to appear, he could show there was nothing wrong with the meat, there was nothing wrong with the jar. Everything was working as he expected. That's what a control is for. A control shows you that your experiment works the way you expect it to. So any control in an experiment exists to let the experimenter verify that things are working the way they expect. And that's all a control is. So that was pretty compelling evidence of where maggots come from, long before we had the microscopes and things that we might be able to use to watch a fly laying eggs. This was before anyone had seen the eggs, the microscopic eggs laid by flies. So the next thing we look at is Antony van Leeuwenhoek's fantastic microscope. He had access to compound microscopes, but they didn't have the magnifying power you need to see microorganisms. And part of the problem was that glass manufacturing was not very good. So what he did was make his own lens. His own lens had a magnification of around 250 times. So a very strong, tiny little lens. And he put the specimen on the head of this pin. In fact, what he did was he just scraped his teeth with this pin and looked at what he found between his teeth. And he reported seeing animalcules from between his teeth. That's what he called the microorganisms he saw. And when he looked at meat, he saw tiny eggs. And these would have been the eggs that would become maggots. These would have been the eggs laid by flies. So Leeuwenhoek's microscope gave us information we could use to understand where flies come from, where maggots come from. And also, this was the first evidence of the existence of microorganisms. So where do the animalcules come from? That's a question lots of people would have wondered is, do these microorganisms, do they spontaneously appear? Do they come from some kind of corrupting gas? Do they come from some electricity? Where do they come from? And several different people tried to do experiments to show either yes or no, they spontaneously were generated. So one of them in 1740s, Needham, he thinks that animalcules arise from a vital force. So what he wanted to show was that this vital force was mysterious, and he could take an non-corrupted material and it would become corrupt. So for example, what he did was he took mutton broth. So this is, you put, I think, lamb's meat in water, boil it, and whatever is in the water after you've boiled it for a while, that's mutton broth. And that could be used to grow bacteria. So if you put bacteria in mutton broth, they will grow and reproduce, and it will start to smell bad. It will rot. So what would we predict if we were to boil mutton broth and then seal the tubes? This is what Needham did in his experiment. So what would we expect, and what would we think is going on? And I want to draw this out for you before we get into the rest of the experiments. I want to make sure we're all on the same page. So I'm going to get out my very low-tech drawing tablet and draw some things. Okay, so let's think about what goes on inside a tube full of mutton broth. So this is a tube, and I am not good at drawing. Inside the tube, there is some liquid, so that's what this would be. This is like some kind of test tube. And the idea, what we now know is that what we now know is that there are bacteria everywhere. There are viruses everywhere. Anything you get in nature, anything you get inside or outside or anywhere is covered with bacteria unless you have specifically destroyed them somehow. So mutton broth would have lots of little cells in it. And when I say lots, I mean enough that it mattered, but not so many that you could see them under a microscope. If you looked at mutton broth under a microscope, you probably wouldn't see any bacteria just because it's a numbers thing that I could explain to you if you're curious. But there would be some in there, and you just have to get really lucky to find them under the microscope. But the idea is, if you then incubated that mutton broth, what you'd get after a certain amount of time, see how bad I am at drawing? Each of these little cells would have reproduced and made a daughter cell. So you'd have twice as many. And then a short time later, all those daughter cells would reproduce, and you'd have 4 times as many. And basically, eventually what you have is just a cloudy liquid that is full of these cells. Okay, so what is the idea with things like stoppering the tubes? Well, the idea is Needham was going to boil one of these tubes. So he was going to use, fire to heat up the tube. And the idea there is that it would kill any microorganisms that were present. And then he would put a stopper in the tube and seal it so that no other microorganisms from the air or from dust particles or from anything else could get in. That's his idea. So his expectation would be there are no live cells in the mutton broth if it's stoppered. And we do this in a microbiology laboratory all the time. We will take a liquid. If we want to use it for growing bacteria, first we kill all the bacteria inside it. We sterilize it using heat. That is a very common thing we do. And if we then seal it, it will stay non-corrupted. It will not rot. The only cells in it would be dead. That's what we know will happen now. But what he thought was he could kill these cells and there'd be dead cells in it after burning it, but then some kind of vital animating force would enter the tube and generate more cells to replace the ones that he killed through boiling. That's what he thought. And so then you'd end up with a rotten tube. So that's what Needham thought he could do, is he would boil a tube of mutton broth, kill any cells that are inside it, and then a vital force would come in and reanimate them, and the liquid would rot. That's what he thought. So what actually happened? What actually happened? So I just walked through what we would predict. But what actually happened was his tubes rotted. So this is what happened. Or I should say, this was how he started, and this was the end, and in between, something happened. So what happened? And that's what we're going to start with in the next video. We will come back to Needham and we'll look at an experiment that helped to show what was going on with this.